Welcome everyone to today's discussion that takes us to the world of transgenic animals. This video clip includes from glowing mice to bioengineered livestock. Transgenic animals are creating the innovations in the field of biotechnology, medicine, and agriculture. What are transgenic animals? Basically, transgenic animals are animals whose genome has been altered by the addition of foreign DNA from other species. This can be done to improve genetic traits like increased milk or wool production in animals. Common methods to create transgenic animals include microinjecting DNA, retroviruses, and embryonic stem cells. The primary and specific goals of creating transgenic animals. The first one is medical research to study human diseases, develop new treatments, and test new medicines. In medical field, basically transgenic animals can be engineered to mimic human diseases, can also be used to produce human-like organs that can be used in diagnosis, treatment. The next one is agriculture to improve crop yields, disease resistance, and nutrient content. In the field of agriculture, the transgenic animals such as insects or mice can be engineered to produce pesticides or other compounds that control the pests that damage the crops. The history of transgenic animals dates back to the 1970s and 1980s when scientists first began experimenting with genetic engineering techniques. The very first transgenic pig was also created in this phase of the history of transgenic animals. Next phase in the history of transgenic animals is the advances in technology. This phase was from 1990s to 2000s. The creation of knockout mice, mice with specific genes disabled, revolutionized the field of genetics and disease research. The question arises, what is knockout mice? Knockout mice is basically is a type of transgenic animal that has been genetically engineered to have a specific gene knocked out or inactivated. The third phase in the history of transgenic animals is the contemporary research. This phase was from 2010s and present till now. The development of CRISPR-Cas9 gene editing technology has made it easier and more precise to create transgenic animals. CRISPR-Cas9 is a bacterial defense system that has been adopted for gene editing, gene therapy, cancer research, and synthetic biology. Here is a brief timeline of transgenic animals that start from 1970s and present till now. In 1973, DNA recombinant technology was developed enabling genetic manipulation. 1980, first transgenic mice are created by transferring a gene from one species to another. After the creation of first transgenic mice in 1985, first transgenic pigs and sheep are also produced. 1987, pro-nuclear microinjection technique is developed improving gene transfer efficiency. This technique is also known as DNA microinjecting. 1990s, somatic cell nuclear transfer (SCNT) technique is developed allowing cloning of mammals. Dolly sheep was created using this technique. In 1996, Dolly the sheep, first mammal, was cloned. This was cloned using somatic cell nuclear transfer technique. For this purpose, scientists collected somatic cells from an adult mammal. The collected cells were grown in a lab dish where they multiplied to form cell culture. The nuclei then extracted, including DNA from this cell culture. These nuclei then transferred to enucleated egg cells. The egg cells were stimulated to divide and then planted to surrogate mother. Then surrogate mother gives birth to a lamb which was named as Dolly the sheep. In 2000, DNA interference technology is developed enabling gene silencing. 2010, CRISPR-Cas9 gene editing technique is developed revolutionizing precise editing. 2014, first CRISPR edited animal mice are reported. Until now, present day, transgenic animals are widely used in research, agriculture, and biotechnology. The methods of creation of transgenic animals. The first one is DNA microinjection, introducing the transgenic DNA directly into the zygote at an early stage of development. In this method, no any particular vector is required. The second method of creation of transgenic animals is retrovirus mediated gene transfer. In this method, infecting mouse embryo with a retrovirus which carry the new gene, using virus as a vector in this method. Applications of transgenic animals. The first one is glowing mice. Scientists inserted a gene from a jellyfish into mice, making them glow in the dark. This helps researchers study gene expression and track cancer cells. The second one is spider goat. Researchers introduced a gene from a spider into goats, enabling them to produce silk proteins in their milk. This could lead to the production of stronger, more durable textiles. The third one is fast glowing salmon. Researchers inserted a gene from a Pacific Chinook salmon into Atlantic salmon. that enables it to grow fast. In conclusion, we can say that transgenic animals are genetically engineered to carry the desired gene from another species that create innovations in the field of biotechnology, medicine, and agriculture. I hope this discussion has provided you with valuable insights into the transgenic animals. Thank you for listening, and I look forward to your feedback. Have a good day.